triple mutant. So this triple mutant will lead to produce this compound. Then this NNO linkage is quite a unique feature in natural product. Then this example of the naturally occurring azokishi alkane. But at this stage, every researcher could not determine why NNO linkage is constructed by enzyme or non-enzyme or non-enzymatic process or not. Therefore, we focus on this biosynthetic machinery. Then, so first we focus on the biosynthetic origin. Then, so second will be analysis of the machinery. Then, this is the summary of biosynthetic origin. This one. So we fed deuterium or C13 labeled compound into the producing strain. Then so we determined this part is from the hexylamine. And so this nitrogen is from the, now it is from the glutamic acid. And so this nitrogen and this C2, 3, 4, this 3 carbon skeleton is from the serine. Then green one, C1 position of the carbon is from the acetate of C2 of acetate. Then so this research will be published in ChemiBiochem. Then so, but at that time, so we have not determined the origin, why NNO azokishi linkage is produced. Therefore, we focus on another azokishi alkane compound. Then so we analyze the genome sequence of other azokishi alkane producer. Then so we now focus on these genes. These genes are common in azokishi alkane biosynthetic pathway. Therefore, we will try to activate all genes to detect the, for example, this AZX VLB, this will be mutation could not produce any azokishi alkane compound. It means this VLB is responsible for production of azokishi alkane. Therefore, so this kind of extensive mutation will lead to predict the azokishi alkane biosynthetic pathway. This will be NNO bond formation. Every researcher in the world do not be determined in this pathway, this one. And so we'll try to be first investigator in this unique azokishi alkane compound biosynthesis. And so we also analyzed the biological activity of this azokishi alkane compound. And so we found that quorum, these kind of K57 and manumycin has a quorum sensing inhibitory activity. But this inhibitory activity is not so strong. But I think they have activity. For example, K57A has a 500 microgram per milliliter inhibitory activity. I think this quorum sensing is used for this N-acetyl formylserine lactone, and for example, in the pseudomonas or something else. And I think chain length will be similar to their compound. Therefore, so it may be act as a inhibitory, competitive inhibitor, I think. But yeah, mechanism is not well characterized. Therefore, so we will try to synthesize or by chemical synthesis or enzymatic synthesis or not. But anyway, so we will try to figure out why these compound has a quorum sensing inhibitory activity in the future. The fourth story, this finding is carried by Dr. Yoshi Nindita. So she, our group found mutation of the plasmid. Then so the plasmid-less mutant could not contain any terminal region of the chromosome. It means the loss of linear plasmid, PSL2L and M, will result in a chromosome deletion. So she found that TPG tap gene pair on the linear plasmid function to maintain the linear topology of the chromosome in such methodology. So this result was published in Molecular Microbiology in 2015. This finding is her Ph.D. thesis. I think she will try to about this story in detail. Then now, we focus on the deletion region of these three plasmid-less mutant. And this morphology, and the production also analyzed by Yoshi and Amiruddin Fauji, Ahmad Fauji. So he focused on the biosynthesis of this compound and analyzed the chemical structure of this structure. And so he isolated several compounds from these three plasmid-less mutants. And so some of them will be commercially available, but anyway, so he found out this compound. But this compound is already identified by other research groups, and so their activity is also identified. But at that time, so we have not yet determined why this compound is produced through NRPS pathway or amino acid TRA utilizing enzyme. Therefore, so I think he tried to analyze the device machinery in his master course student master thesis. So, but if he can promote it as a next scholarship, so he will try to continue this work or other research in my laboratory, if possible. Yeah, it depends on next decision. Then I will talk about the fifth story. So as I mentioned in the first topic, this compound, salt-methane signal molecule, is activate secondary metabolite production at nanomolar concentration. Then we focus on this kind of signal molecule as a microbial communication tool. So this signal molecule has several merits. First merit is no need of genetic engineering. So at that time, so strategy 1 to 4, so they require genetic engineering. In that case, it's difficult. For example, if we visit some area, it's difficult, perform the genetic engineering in a university or in nature. Yeah, but this will be no need to genetic engineering. Just the addition of this compound will be enough to obtain the silent secondary metabolite gene cluster activation. Then the second merit will be small amount of compound are enough for genome mining, I think. But we performed this compound will be available for the microbial communication tool for activation of the silent secondary metabolite gene cluster. This concept of this genetic engineering-free genome mining approaches. So as I can say, I talked in first topic. So over 30 biosynthetic gene cluster for secondary metabolites are located on the straight message genome. However, in normal culture condition, only 10 to 20% of metabolites are produced. For example, in this figure, only 4 compounds are produced. But so if we add this signal molecule as a chemical inducer, then so if this compound will be responsible for activation of the biosynthetic gene cluster by addition of the chemical inducer, so this blue one could be, if this compound be induced by chemical inducer, it will be good for genome mining approaches. But in some cases, I think diminished production also will be detected. Because if these compound is accumulated and by addition of this compound, maybe some of them will be diminished due to the use of a precursor for secondary metabolites. So anyway, so we will try to investigate the signal molecule can act as a versatility inducer for metabolite production. So these our pilot screening for genome mining approach of addition of a signal molecule. Then, among them, so we found that change of the metabolic profile in among 69, only around 8 strain among 69 strain. So at that time, so we have not determined the chemical structure of this, what kind of compound are activated or not. Anyway, so we will try to investigate in detail and in the future. Then these inhibitory assay. Then so we found, addition, for example, the addition of the signal molecule will activate anti-inhibitory activity here. And some of them inhibit addition of the antibiotic activity. But anyway, so any change in inhibitory zone will be found in 11 strain. So these show summary of the activation site secondary cluster. So these 5 strategy well performed in our laboratory, and so I will try to get a novel natural product from the streptomyces genome. Then, okay, finally, so I will talk about other topics. It's kind of an international research collaboration. And so this will be finally, maybe next month, this program will be finalized. But research will be continued from now on. So we submitted JSPS SDDF bilateral research program. We accepted in structural redesign and computational simulation of rancacidin analogs to improve anti-tumor activity. So he's an Egyptian, Ahmad Ayoub. So he's an expert in computational chemistry. Then so this is my slide, and so okay, this is a concept of our research collaboration. Then so initially, so we at first we found a collaboration, and we found that this is rancacidin, a compound we are talking about figure out this biosynthetic pathway. And so he found that this compound may act as an anti-cancer agent by computational prediction. So he discovered this kind of a compound will be effective, most effective agent, anti-cancer agent, by computational simulation from the 3 million computer chemistry compound databases. However, this compound is just a prediction, so it's difficult to obtain this compound only from the chemical synthesis. Therefore, so he asked me to give this compound. And so this 3D overlay of rancacidin and with the molecule, this compound. Then they are made a good fit with each other. And so finally we found that this rancacidin also has anti-tumor activity. Looks like a paclitaxel-like action. It means beta tubulin stabilizer for action of the anti-cancer activity. Then, so this is a concept of our research collaboration. Then, so we will try to modify the chemical structure of this compound. Then, so based on their computational simulation. Then, so if that compound has anti-tumor activity, then so Egyptian side will modify the chemical structure by computational simulation. And so they're candidate for compound with improved anti-tumor activity. If it works well, so we will try to modify the structure by organic synthesis or enzymatic bioconversion. Then so if this cycle well good, so finally so we regard the ultimate anti-tumor compound. Then, okay, I will talk about the 3 Indonesian in my laboratory. First one, Dr. Yoshi Ninjita. She graduated Bandung Institute of Technology, and so she came to my laboratory in 2007, right? Yeah. And then so she got a master thesis and a PhD thesis in my laboratory, and so she moved to Oregon State University as a postdoctor for 1 and 6 months, 9 months, I think. And so I think her story we talked by Yoshi. Therefore, so please listen to her presentation. Then, so second Indonesian is Amiruddin Ahmad Fauzi. He graduated this University of Jember. Then so he came to our laboratory 2016, and so he got a master thesis in 2018 September. Then so he tried to extend his PhD work in my laboratory. However, as you know, LPDP changed the regulation. Therefore, so he has to return to Indonesia. Then so now he work on something else. Then I think he will try to apply again, I think. Yeah. And so third Indonesian is Lukman Muslimin. So he graduated in the University of Negeri Makassar in Sulawesi Island. And so he also graduated in Hiroshima University in 2019 in March. Yeah. Then so in my present visit, so we met both of them, and also in this time. And so our group focus on international. We attend the international meeting. So 2017, so we visited Jeju Island in Korea. So Yoshi and Amiruddin Fauzi here. And so next year, so 2018, in July, so we went to Sapporo, Japan. So Amiruddin is here, and Lukman is here. So Yoshi was also already went to Oregon State University. She's not here. And so yesterday, so we took a picture with Amir here. And so in front of the so monument of the University of Jember. And so I hope their progress in their mother country or other country is my treasure. Yeah. And this slide, okay. Can you find Dr. Adi? Here. Okay. This picture we take in 47 kilogram. He looks very smart. Yeah, here. Yeah, but Dr. Adi is in other laboratory, but same floor. So we met, many times in per day. Therefore, I think he's quite a hard worker in Professor Yamada's laboratory. And so I hope so my 3 students will be good, successful in like Dr. Adi in your future. Yeah. Yeah, looks smart. Yeah, and let's be now Hiroshima University has 1,900 international students. Among them, so Indonesian is 156% students. Maybe second position, I think. Yeah, first one is over 1,300 from China. Then second will be Indonesia. Yeah. Therefore, I think there's many Indonesians. So I think it's good for communication with Indonesian students in Hiroshima University. Then, so our group focus on biosensitive pathway of this natural product. But So we extended our research is now expanding. For example, microbial resource, from microbial resource, they purify the chemical structure and elucidation and the genome mining approach to obtain the natural product. It means the natural product discovery. Then, we also focus on the chemical eliciter, for example, signal molecule. And next, chemical natural product could be converted unique biological function. It means the improvement of biological active function and the rational molecular design of the highly functional bioactive substance by either organic synthesis or structural modification by enzyme. Then, but in our group has no technique on the isolation of unique and novel bioresources and discovering the unique biological activity. We just performed on the antimicrobial activity and fungal activity. It's a normal antibiotic activity only. Therefore, discovering the unique biological activity was responsible for our expanding our natural product discovery. Then, I will show you this kind. We collaborate with this professor. Then, last year, we got a JSPS Kakenhi. It's a kind of a joint international research program. Then, we started a joint research program with University of Negeri Makassar and our Hiroshima University and the Tokyo University of Marine Science and Technology. Discovering a unique biological activity, will be carried by Dr. Kuniyoshi and Dr. Koyama. Their specialty is moon jellyfish and shrimp. Then, I think we will try to obtain some unique biological activity using this kind of organism. And Dr. Ali is expert in isolation of unique and novel bioresources from the here, Makassar, Sulawesi. Yeah. Then, in this project, we focus on responses against global warming. As maybe Adi and Yoshi know, Japan has 4 seasons: spring, summer, autumn, and winter. They are independent seasons. But I think global warming will lead us to maybe tropical area. Therefore, in that case, it's difficult to form the tropical specific, for example, diseases or something else. We have no resistance to. Therefore, this project will be good for healthy aging or something else. Then, this project is also good for. We already contracted the collaborative research agreement, including ABS and the university cooperation. And I think he will try to find this kind of bioresources from the island and karst. Then but we just started. Then I think I'm focused on any overseas researchers, especially in Dr. Adi also. And we will try to ask someone unique kind of novel biologists, and especially in Java also. I think I'm really interested in expanding our research responsibility, collaboration responsibility. And finally, our natural product discovery will be good for contribution of healthy aging and the drug repositioning. It means the redefinition of drug. And 3 years ago, we found the anti-tumor activity of lancashide. This compound is well known as anti-microbial activity, but we evaluated the anti-tumor activity. Yeah, it's kind of a redefinition of the drugs. And recently, coronavirus is terrible problem in the world. Therefore, as I heard, anti-HIV agent is good for against the coronavirus. And so if our research collaboration with drug repositioning will lead to a great activity agent for healthy aging or something else, and human health also. Then CCB acknowledgement. So these three Indonesian students worked with me, and so they performed many experiments and published in something else. And so it's final. So thank you for your consultation.
Thank you, Professor Arakawa. This is a very interesting and quite advanced lecture today that I hope and I believe all the audience here understands and then understand about the lecture today. Do you agree with me? Ini presentasinya sudah bisa dipahami semua ya? Bisa dipaham kontennya? Sudah dipahami? Saya yakin sudah dipahami. Kalau belum paham, bisa memberikan pertanyaan. Mungkin before we start the discussions, you may have a seat back. Okay, I have some notes here about the presentation today because according to the title here, the microbial nature products discovery through various genome mining approach. So I remember about how Alexander Fleming found the penicillin. So after he found the penicillin, he tried to make it more mass productions in industrial way. And then, according to this lecture today, we found that two mass productions and such antibiotic or secondary metabolite product from the microbiome is not easy because according to Professor Arakawa presentations, only 10 to 20% of microbial can produce the secondary metabolites. So it's not easy way. So it's why the Professor Arakawa group tried to find the strategy to boost the production of secondary metabolites through, I noted here about 5 strategies to boost the production of secondary metabolite, including the antibiotics. So these antibiotics are very useful, and here is very interesting one, and eye-catching is anti-HIV agent against the COVID-19, the very terrible issues today in the world about the coronavirus. So this is very perspective issue for today and for the future. So I hope you may give feedback to Professor Arakawa with your questions, which parts that you don't understand or you need more clarifications, maybe about the functions, why you do this such experiment, what is the function of the products for the public. Okay, for the sessions, I will open like 3 persons to give a questions or any request for clarifications. Okay, 3, 1. We have 2, Professor C. Agus, Dr. Nurhayati. We have last one. Any student? Okay, so 1. I guess Fitri? Okay, for the first question come from Dr. Nurhayati. Please, if you have.
Bismillahirrahmanirrahim. Thank you for this opportunity. Bapak Arakawa, my name is Nurhayati, come from Biotechnology Magister and Department of Agricultural Product Technology. This is a great presentation from you. So it is a good news for us about the challenge anti-HIV agent, anti-COVID-19. My questions, what happened, what happened about streptomycin, where we compare with the others antibiotic. Maybe streptomycin have large spectrum for the antibiotic. Please inform us about the spectrum action for the antibiotic compared to the other antibiotic. Because based on your research, that you focus about the streptomyces. We know that streptomyces is positive gram bacteria, and this is a spore bacteria, can produce the spore. So I think this is more survive to the challenge environment. So we can modify the strength environment for produce the others metabolites. So my second question about why we can compare the antibiotic with the anti-HIV, because we know that antibiotic can destruct the bacteria, while the HIV is this from virus, and the virus change our immunodeficiency. But for antibiotic, this is about the bacteria or the pathogenic microbes. Please give me more information about the challenge antibiotic with antivirus. Thank you very much.
Okay, thank you, Dr. Nur Hayati. Next question from Professor Triagus, maybe one or yeah,
it's
at
very
least one.
simple question, maybe, Sensei. I have question, but before that, I would like to thank for nice lecture today. And then from your lecture, you introduce to us how to strategies to produce benefits compounds from silent gains using genetic engineering. And then you have many strategy to produce some benefit compound. I have a simple question: how to initiate construct work to make strategies? Just that's enough. Thank you. How to initiate construct work? Yeah. Because you know that the strategy not only to genetic engineering, but the environmental depend with the environmental also. Okay. Thank you.
Okay, I get the question, so I will repeat the question later. Okay, and the last question for the first sessions, Ms. Fitri.
Assalamualaikum warahmatullahi wabarakatuh. Hayo gozaimasu. My name is Fitri from Chemical Engineering. Kagaku no Wagaku. So I'm interested with this new metabolite, but because my subject is chemical engineering, so I'm working with optimization. So I would like to ask, what is the problem actually? Maybe similar with Dr. Nur Hayati's question. How can we optimize the system to make it more conducive for the bacteria to produce the metabolites? I'm interested with this? And the second is, have you ever confirmed that the metabolites does not cause resistance to the virus or to the bacteria, other. ありがとうございます。
Thank you. Okay, now we have 3 persons who ask you maybe about like 5 questions we have. So, would you like to answer all together or one by one? Okay, the first question from the Dr. Nuri Hayati, because the first question is, what is the difference of the antibiotic of Streptomyces to other bacteria? Because we know the bacteria, almost all bacteria can produce antibiotic, but in this case, you focus on Streptomyces. Why? So what is the difference between the antibiotic produced by Streptomyces and other bacteria? Maybe the first question. The second question, this is related to anti-COVID. So the anti-HIV agents against COVID-19, because she imagined surprised because this is antibiotics. So antibiotics normally against the bacteria, but not the virus. So how can you explain for example, here Lancacidins has a prospect or potential to be anti-HIV agents against COVID-19? Okay, this is from Dr. Nuyati. Maybe you can answer first. So later I will repeat again the question from Prof. Triagus and Dr. Fitri.
Okay, thank you. Okay, your first question will be, yeah. So, yeah, antibiotics will be produced by many bacteria and fungi also, right? But in my laboratory, in my previous supervisor focused on Streptomyces. Then, so most important advantage in Streptomyces handling Streptomyces is easy to handle the genetics. Yeah, it will be well established by England research group and John Innes Research Institute. And so therefore, it's easy to manipulate their genetic engineering. Then, so we also focus on other bacteria by genetic engineering, but it takes more time, to handle. And so, yeah, I think in near future we will try to, perform the genetic engineering with other strain, other species. Yeah. Then, second question. We have not yet, determined, so our compound has an anti-coronavirus or not. But I think the mechanism is quite difficult to predict because, so in 2016, we found the anti-tumor activity of Lancacidin. So now we understand this compound could bind to beta tubulin. Beta tubulin is responsible for mitosis of the cell division. Then, so it means the anti-cancer activity will be performed by this compound. But I think it's totally different with the antivirus and antibacterial. But we have to try, to perform this, So any already known compound could responsible for CDVID 619. Yeah. In that case, it may be good for our future. Yeah. It's difficult. Yeah.
Do you get the answer? So at least for anti-COVID, this is still prediction. So this is because based on previous study, it's like has potential to combat or to fight the COVID in the cells. Okay, the third questions from Professor Tri Agus, how to initiate the construct of overall work, because you already have a 5 strategy, how you initiate it?
At first, yeah, most easiest way would be focus on a repressor, yeah, because it's easy to just to focus on the repressor. Then so it will be available for other streptomyces species and other microorganisms, I think. Yeah. And so we extended the 5 strategy. So many my friends in Japan. So why you use this many strategies to focus on these streptomyces? But I will try to discover many compounds from the one strain. It be because I extensively focus on this strain. Therefore, so we try to plenty of the strategy. Then so I know this strain. Everything in this strain. Maybe Yoshi also knows this strain. Then so I hope we will try to improve their strategy to discover compound. Then so I think these 5 strategy will be responsible for other species and also So I think a combination will be good for obtain the compound. Then so occasionally I found in strategy 3, so we found a unique compound from our strain. So it's quite difficult to select.
The last 2 questions from Dr. Fitri. The first is, what is the problem of the optimizations of the production of secondary metabolites of streptomyces? And then the second questions, if the secondary metabolite is antibiotics, because we know the antibiotics sometimes trigger the resistance or the immunity of the bacterial cells to be resistant to such antibiotics. So does the product of the secondary metabolite in your research affect on the resistance?
Okay, first question. Okay, thank you for your question. So in my your first question, so yes, streptomyces has advantage to produce many antibiotics. However, in some cases, it's difficult to obtain the large amount of the compound. Therefore, so some industry will ask us to improve the antibiotic production by genetic engineering or something. And so I think strategy 2, manipulation of the repressor will be effective. But especially in Japan, so manipulation of genetic gene in microorganisms, well, consumer don't want to use the genetic and modified GMO, it looks like GMO. But other strategy 1 to 4 will be oral GMO. Therefore, so now we focus on the strategy 5, but not the GMO. Yeah. So.
Arakawa, before you continue to answer the second questions, because in your first presentations, it's mentioned that only 10 to 20% metabolites, secondary metabolite produced in normal conditions. Only 10 to 20%. what is the problem? In the normal condition, they produce a low amount of secondary metabolites.
Okay, I see. Oh, I think I say the normal condition, but it is isolated strain, right? So in environment, they have many other species in soil. Then I think they may produce other compound in soil, but we have not protected, so in a mixed conditions, I think they may produce something, yeah, but it's difficult, I think maybe combination will help us to predict why they only produce 10 to 20%. I think other metabolite could be produced in mixed condition, it looks like in soil, but we are not sure. Yeah.
And then what about the resistance?
Yeah, it's also difficult question.
You test before the resistance of bacteria against these secondary metabolites, or do you have
not,
any idea?
Not yet. Yeah.
Okay. This can answer your questions. Okay, thank you. Nowadays, these sessions, the first session has been complete, so we move to the second sessions. We give a chance to the student first, and then come back again to the staff. Okay, to the lecture. Okay, 3 students, please. 3 students. Oh, we have new students. Okay, please.
Okay, thank you. My name is Selma Sulistianingsih. I was a student. Okay. I think this slide is really interesting since you also write you are in the research of, on anti-HIV agent against COVID-19, since we know that right now COVID-19 is getting our attention. The world give really a big attention to this virus. And we know that HIV and COVID-19 similar structure. They are both single-strand RNA virus. And what have you found about this? You working with the antivirus and anti-HIV agents and working with the COVID-19. Maybe you can show us, have a publication on that? And also the second question is, are you also working with the diagnostic kit for COVID-19? Since right now our government buy a primer from Japan, but I don't know from where, maybe it's from your lab. Okay, so thank you.
Assalamualaikum warahmatullahi wabarakatuh. Oke, selamat pagi. May I ask in bahasa Indonesia? Oke. Jadi saya mau menanyakan. Jadi nama saya Ahmad Fatihul Hasan. Saya mahasiswa dari program studi ilmu pertanian. Saya konsentrasi di ekonomi pertanian. Nah, yang ingin saya tanyakan sebenarnya terkait dengan beberapa frasa terakhir yang diucapkan oleh Pak Arakawa, jadi terkait dengan bahwa masyarakat itu tidak terlalu menerima GMO. Nah, yang ingin saya tanyakan adalah terkait dengan asumsi dasar dari penelitian bioteknologi, bahwa bioteknologi itu ingin menghasilkan, kalau dari penelitian ini adalah untuk obat yang karakteristiknya itu misalkan anti tumor, obat yang punya obat hasil penelitian bioteknologi dengan kapabilitas anti tumor. Nah, saya juga meyakini dari keberhasilan pengembangan penelitian bioteknologi adalah ketika produknya itu digunakan secara luas oleh end user atau masyarakat. Nah, yang menjadi perhatian adalah bagaimana jika asumsi dasar keterterimaan produk ini salah. Misalkan kita mengembangkan obat tumor dan asumsinya adalah obat tumor itu akan diterima ketika kekuatan antitumornya tinggi, akan diterima masyarakat ketika antitumornya tinggi. Nah, asumsi dasarnya adalah masyarakat menginginkan obat yang antitumornya tinggi. Nah, bagaimana jika asumsi dasar tersebut salah? Atau paling tidak itu bukan karakteristik utama yang diinginkan masyarakat. Bagaimana ketika kondisi terjadi seperti itu? Dari sisi penelitian, apakah itu akan secara signifikan merubah arah penelitian yang sudah ada? Jadi itu yang ingin saya tanyakan. Terima kasih, selamat pagi, assalamualaikum warahmatullahi wabarakatuh.
Okay, thank you. My name is Rido Firmansyah from Biotechnology student. I have two questions. The first question is, for us, the researcher, if we want to mining such organism or microorganism, let's say another organism, which mining method do you want to suggest to us? Which the method is the best method for us if we want to newly mining the new organism? From your discussion, we already know about 3 methods you have explained before. Which method do you prefer more to us to mining such of new organism? And my second question is, from your finding or the result of the antibiotic, it is perform well in their current form, or it will need to be reconstructed or modified again to perform in a better performance? Thank you.
Okay, answer to first question. So maybe anti-virus agent, maybe COVID-19 is, I have noticed in a microscope, but so in TV or internet, so they will see shape and something behavior symptom will be quite similar to SARS, SARS, yeah. So I think SARS is effective, so be treated by anti-HIV virus. Then so my friend in Japan also talked with what kind of compound will be good for treatment of COVID. And so I think antivirus anti-HIV agent may be good for this kind of treatment. But so in streptomyces, not only streptomyces and fungi. And so I think there are many secondary metabolites. However, so it's difficult to obtain the antivirus compound, antivirus agent. Therefore, I think we will try to focus on not only for antimicrobial activity and cancer. Therefore, we will try to we have to find out so antivirus activity, yeah, in near future. I'm not sure. Yeah, maybe so. Yeah, maybe so. Lancasizing would not have such anti virus activity, but we have not yet tested yet. So I think we will try. We can try. Yeah. 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 So in our five strategy, so many people focus on genetic modification. Yeah. And so now we tentatively focus on non-GMO approaches. So I'm not sure whether so it works well or not, but just a trial, yeah. Maybe GMO will be, I think genetic modification will be more effective, you know, maybe targeting, isolation of the compound from the species. I think it's a good tool for obtaining the bioactive substance from the species strain. Yeah.
Okay. In your case, why you choose non-genetic engineering way to improve your product than using the modifications? Why in your case you choose strategy? Because in strategy non-genetic engineering, you can. Why you choose this one?
35, right? You mean?
In the case of your strategy, you have 5 strategies.
Yeah.
So among them, one of them is non-genetic engineering.
Yes.
Non-genetic.
Yeah.
Why you choose this one, not genetic one,
yeah.
genetic engineering one?
Yeah. Okay. In strategy 5, so I think I don't know. So we isolated the signal molecule. It looks like a chemical inducer by ourselves. Then so we want to improve our potential to that compound for microbial communication. Then, so I think in soil, so there are many species works on each other, and so I think some chemical substance, including and it's kind of chemical inducer may initiate their independent strain. Then so we focus on that compound. Yeah, I think it's maybe lower amount to be effective. Then so that compound has no antibacterial activity. So it may be advantage. I think in soil, so it's some kind of antibiotics may works as a communication. But in some strain, maybe that, for example, antibiotic A will be sensitive to strain B or something. In that case, strain B will be passed away, right? Then but this compound has no antibiotic activity. Therefore, so I think it's good for microbial communication. But yeah, now so many researchers focus on it's kind of a microbial consortium or communication. Yeah, therefore we focus on that one. Yeah, but yeah, I think. But at least 10% of strain could be activated by addition of the chemical inducer. So I will try to, yeah. Yeah, yeah.
Okay. Additional question also from the same persons. In your presentation also, you mentioned about the anti-tumor activity of lancacidins. So based on the research in the laboratory, you found that this has potential as anti-tumor. But what happened if after mass productions and then released to the public and then they try and then actually this is too high or very low activity? So what is your idea? You should change the experiment or something?
Yeah, maybe we will perform the, so analyze the biological activity, my experience. Then, so in this collaboration, so I think they just focus on the computational simulation, right? Then, but their computational chemistry, so they don't know, so the chemical modification is quite a terrible task, For example, that compound has several hydroxyl group. Then, so it's difficult to, target then protection where modification with only one hydroxyl group. Then, in some cases, it's difficult. I think further modification will be maybe quite terrible and quite difficult to modify. But yeah. Anyway, so we will try to simple modification may give us great information to improve the anti-tumor activity. So now, so anti-tumor activity was increased over tenfold. But in paclitaxel is used for anti-tumor agent. But its side effect may be it may have a side toxicity. Then, so if lancacidin is not toxic, and if we improve the anti-tumor activity, it will be good for Yeah.
Did you get your answer? Because in this case, Professor Arakawa doesn't use genetic modifications as a strategy, but instead of boosting by adding additional or modification environmental, not the genetic, to produce secondary metabolites. So maybe, and also according to anti-tumor, this is still in the computational experiment, not proof in the real experiment using the cell or something. So they predicted that such kinds of secondary metabolite has potential to be anti-tumor, but need more proof using the experimental cells. So, okay, the last question from Rido, Mr. Rido, Mr. or Mr. not on Miss Mistress, Mr. not Miss. Okay, Rido has a question. What is the best method to start the mining of new strain in case of
Okay. I think best strategy for genome mining will be strategy 2, inactivation of the repressor. But so in what we need to perform the genome sequencing, yeah. So if we got over 100 strain, so but we have to do 100 genetic sequence. Yeah, in that case it's quite takes time and cost. Yeah, but I think most effective will be strategy too. Yeah, in activation of a repressor. Yeah. Yeah. Ah. Yeah. Yeah. We have to improve the strategy, our strategy more. So if we have plenty of from the soil, then I think as I mentioned, so genetically sequence should be required. In that case, I think strategy 5 is easy. Yeah. Without any information, just addition would be good for okay. But at this stage, we have not practical evidence, just a pilot screening. So if we obtain the practical result, so I think we can we have a confidence to this strategy is good for large many sample, yeah, for screening. Yeah.
In case of your streptomyces, how many gigabase pair or megabase pair of the sequence of the genome sequence?
One strain.
And then Rohi or something.
Yeah.
Yeah, you're saying how many base pair or megabase pair the genome size?
The genome size will be 8 megabase. Yeah.
8 megabase.
8 megabase. Yeah.
So we have to sequence first from 8 megabase pair and then finding anything inside and then start to work with the strategy.
Yes.
Bagaimana? Okay, we have like 20 minutes more. Maybe last sessions, the third sessions, 3 questions. First from Dr. Jayus, Dr. Banun. One more, please. Students. Okay, one, please. Okay, I hope students can understand what is the lecture today. Okay, only 2, the last sessions. Okay, for Dr. Jayus, please.
Okay, I'm not a student anymore, but I got a question from my student. Thank you, Pak Rian. The first question from my student is asking about a silent secondary metabolite. Why do you mention that secondary metabolite is a silent metabolite? That's the question from the student. The second question is about the difference between signaling molecules and the versatile inducers. Is that the same thing or different thing? Or is there any difference between signaling molecules and the versatile inducer? Because you're talking about this signaling molecules. And the last question may be similar to previous student. Actually, how to find a measure, find the first method to find a signaling molecule. Because we are going to overproduce a certain metabolite. Is there any fast or efficient method to find these signaling molecules? The third question, you're talking about signaling molecules, and here you mentioned that this signaling molecule is important as a tool for microbial communication. In what aspect, actually, these signaling molecules important to microbial communication tools? Thank you. Okay. From Professor Arakawa abstract here, signaling molecules is induced antibiotic production at nanomolar concentration to evaluate as a chemical elicitors or microbial communication tools. So in what aspect actually that signaling molecules, design the microbial communication tools? Thank you.
Thank you. Arakawa Sensei, I'm very interested with your lecture today, and I only have one question, short question. How the roles of secondary metabolites of streptomyces as interspecies signal in antibiotic production? This is my question. Thank you. The role of the secondary metabolites of streptomyces as interspecies signals in antibiotic production, like this. Thank you.
Okay, thank you. Your question. Why species strain has silent? I think as I talked before, so maybe in this presentation, in the world, so silent means silent in normal culture condition, right? I think in environment, maybe silent metabolites, not silent in some case. We are not sure. Yeah. Then so it's difficult to say. You mean the silent? Why silent metabolites? Yeah. So in genome sequence, the whole genome structure is okay. It's maybe active, responsible for active. Then so, for example, if we extract that gene cluster into the other heterologous streptomyces, it may in my case may be active. Then so this produce something, but in producing organisms, they are silent, no production. I think that in that case, maybe in normal culture condition, in an isolated condition, so they don't produce. Then so in some mixed condition or in specific environments, they may have a potential to produce something. So in genetically, it's okay. Yeah. Yes. Yeah. Maybe they may have it. Yeah. So. Yes. Then, if we transfer to other species in other heterologous expression, in that case it's okay. It means I think whole structure, genomic structure, we okay. Genetic structure, we okay. Yeah. Maybe some condition. For example, temperature or some some induce or something.
Molecule and inducer.
Yeah. In my presentation, we use I use the signal molecule as a word. So it's for strain specific term. And so when I use a chemical inducer or something else, so we are not sure at this stage. Signal molecule is usually for specific strain. But so chemical inducer means global inducer. For example, chemical elaster, looks similar to a chemical elaster. So I just use a chemical inducer, just based on the word, what I use. HA, Homosteric lactone, looks like.
Dan terjawab. Yang ketiga, how to find the signaling molecule.
Yeah. 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 Yeah, it's just our prediction. Just based on the chain length. If we perform this kind of quantum sensing inhibitory activity with larger, more extended chain length, it doesn't work well. So, therefore, maybe we act as a quantum sensing competitor. Molecule competitor of quantum sensing.
Signaling molecule which promote the antibiotic production.
So in genome sequence, so we can obtain the information for signaling molecule specific receptor. It's easy, maybe more than 50% amino acid sequence similarity. Therefore, it's easy to predict. Then, but we have not yet any information, what kind of a signal molecule will, specifically binding. Then, for example, in our strain, we found 7 homologous genes for signaling molecule specific receptor. So one of them is for SRB. And so another 6 we are not known. Therefore, other 6 will be responsible for other signal molecule from the environment or not. Therefore, so that's why strategy 5 may effective for genome mining approaches. But more experiments should be required to elucidate its kind of unique microbial communication tool.
Cukup. Okay. Next from Dr. Banun. So maybe please correct me if maybe I make a wrong note. How the role of the secondary metabolite or signaling? Okay. Signal. Oh, okay. In the production of antibodies. Okay. So, what is the role of the secondary metabolites of streptomycin? That's right. So, what is the functions of the role of?
So, in my in some researcher Professor Julian Davis, he's in Canadian researcher, so he's a famous. And then so he talked to us in why streptomyces produces antibiotics. And so maybe streptomyces or fungi also produces antibiotics to protect their own environment. Yeah, to leave the their own environment. Yeah, to keep them. Yeah, it is one of the possibility. But other researchers said, some strain produces antibiotics to protect their own world. However, the concentration of the antibiotics is not so high. Therefore, I think it's difficult to keep their own environment. So I think some researchers said, why they produce antibiotics. Antibiotics can used for microbial communication. Yeah, and also we don't have any good answer. Therefore, so it's a difficult question. But yeah, main purpose will be to protect their own environment, to protect to survive, from the environment. Yeah.
So, additional questions, and maybe the closing question from me, following up the question from Dr. Banun. So, if one strain of Streptomyces species, in case of Rocky, so they produce secondary metabolite and antibiotics. As you mentioned that this antibiotic will protect this bacteria from the environment conditions against another bacteria. So what will happen? This antibiotic can also inhibit another species of Streptomyces, interspecies. Can antibiotic from Streptomyces rhocae can inhibit Streptomyces caibes or something, or another species? Or do you have any idea?
Yeah, I don't have any idea, but yeah, I think they can protect their own environment. Yeah. Yeah. Yeah, I think so. Yeah. Yeah, maybe. Yeah.
Okay, thank you all questions from the audience, and then thank you for the answer from Dr. Arakawa. We have like 4 minutes, but I think it's impossible to have more questions in this lecture. So I think this is the last session of our lecture today. So actually I want to make a summary, but I have like 4 pages to read, but I think it's impossible to read this paper. But the important things are the notes here, but we have to know that productions of secondary metabolite is not so easy because they need strategy to boost the production. Because in normal conditions, in the laboratory, in the controlled conditions, it's difficult to produce in maximal way. So it's why it still need strategy to boost the productions. But however, the condition in the nature maybe the production still exists and more production compared to in the laboratory. So it's why we have to choose the strategy. In this case, Arakawa and his team has proposed 5 strategy to maintain and boost the secondary metabolite and has potential in anti-aging and then in health and something. Yeah. So I think this is the last notes from me about these presentations, about the lecture today. And I hope this lecture today will be useful and you can give the benefit from this lecture. And don't forget, because pre-discussion with Professor ARAKAWA, he opened for you. If you have any questions, you can ask him by the email. We will give you the email later. And if you're interested to have a joint research with him, he will be. We can organize it in the future. Okay, I think that's all from me. Please give a big applause for Professor ARAKAWA for his presentation. And thank you very much for the presentations. And I close these sessions. Assalamualaikum warahmatullahi wabarakatuh. Thank you very much for your attention.
Thank you, Profesor ARAKAWA. Bapak, Ibu, adik-adik semuanya, saya ucapkan terima kasih atas kehadirannya. Dan insya Allah saya ingatkan sekali lagi untuk mahasiswa program studi Magister, nanti kita ada workshop jam 1 di lantai 2 Sidas, lantai baru, gedung baru. Dan ini tidak bisa menutup kemungkinan jika Bapak, Ibu atau adik-adik yang berkeinginan untuk ikut diskusi di situ, monggo kita terbuka. Ya, di situ ada diskusi interaktif dari Dr. Yosi, ya. Nanti bisa dikomplekan dengan Profesor ARAKAWA. Jika ada yang berminat, silahkan untuk bisa hadir juga di situ. Ya, sekali lagi saya ucapkan terima kasih. Untuk terakhir ini kita coba foto session bersama ya. Ya, untuk mohon Bapak Ibu semuanya untuk bisa ke depan untuk kita bisa foto sama-sama.